an automated electrochemical lateral flow device for one-step hepatitis B virus DNA detection. Nowadays, hepatitis B virus infection is a life-threatening liver infection caused by the hepatitis B virus (HBV), and puts people at high risk of death from chronic hepatitis, cirrhosis, and liver cancer. Globally, chronic hepatitis B affects approximately 257 million people and contributes to an estimated 700,000 deaths worldwide every year. Among these HBV biomarker detections including protein, antibody, and nucleic acid, the nucleic acid detection has been of interest for diagnostic of disease progression. This assay detects particular genetic materials of the virus in biological fluids based on the specificity of the Watson-Crick base pairing rule. As a consequence, DNA detection can be detected at the early stage of infection, which makes it attractive to be used in the clinical area. While various nucleic acid detection techniques like real-time PCR have been demonstrated, their main drawbacks, such as the need for specialized users, dedicated equipment, as well as the time-consuming procedure, limited their usefulness outside the laboratories. To overcome these problems, point-of-care diagnostics offer great potential to detect and monitor infectious diseases in resource-limited settings. Desired characteristics of POC diagnostic technologies including affordability, disposability, portability, and ease of use are required. Presently, the lateral flow assay platform has been widely used as a point-of-care sensing device. The advantages of this platform are rapidness, one-step analysis, low cost, user-friendliness, and portability. However, this platform is limited to only qualitative analysis with a yes/no type of colorimetric detection. On the other hand, electrochemical detection offers both qualitative and quantitative information with excellent sensitivity. Therefore, the implementation of the miniaturized electrochemical detector for eLFA has been of interest. In the field of electrochemical DNA sensors, one of the key components to achieve specific DNA detection is the probe. The pyrrolidinyl peptide nucleic acid, or ACP-cPNA, is a DNA mimic with a peptide backbone which used as a probe to capture HBV DNA. The advantages of the PNA probe are stronger binding affinity and higher specificity toward target DNA over the conventional DNA or PNA probe, which make it interesting to be used in electrochemical DNA sensing platform. In this present work, we proposed an automated paper-based lateral flow electrochemical device for one-step HBV DNA detection. This sensor provides advantages over the conventional method like real-time PCR, including just one-step introduction, 5-fold faster detection time, and enabling for quantifying the HBV DNA without amplification step. The component of eLFA device are comprised of sample pad, wax-printed NC membrane, adsorbent pad, screen-printed electrode, backing card, and upper and lower case. On the wax-printed NC membrane, they consist of delayed channel, non-delayed channel, and test zone. On the delayed channel, the gold ion was dropped on here, while the PNA was immobilized on test zone. Procedures for HBV DNA detection on the sensor. The 50 microliters of serum sample was made up of HBV DNA and PBS buffer pH 7.4 with SDS. The sample was loaded on the sample zone. The solution was moved from sample pad to NC membrane. The HBV DNA was firstly moved along the non-delayed channel, and the solution flow from non-delayed channel were merged and then moved to the test zone. After the solution flow moved to the test zone, HBV DNA will hybridize with complementary PNA probe, while the solution flow from delayed channel is slowly moved to the test zone. Then the solution moved to the end of the channel. The gold ion will be carried to the test zone. Next, when a gold ion moved to the test zone, the binding of gold ion and target DNA would occur via electrostatic force. After that, the gold ion was electrochemically reduced to gold metal with applying -0.2 volt for 120 seconds. Finally, square wave voltammetry was performed to detect the gold metallized DNA via gold signal. The absence of the HBV DNA provided a low current response, while the current response increased with the presence of HBV DNA. All the processes can be achieved within 7 minutes. Results: under the optimal conditions, the current signal of the gold metallized DNA linearly increased with the increasing logarithm concentration of HBV DNA in the range of 10 picomolar to 2 micromolar. The limit of detection is calculated to be 7.23 picomolar. For the specificity, the results showed that the proposed sensor can precisely distinguish from the other DNA sequences, including single-base mismatched, two-base mismatched, HCV complementary DNA and human papilloma DNA sequences because of the high specificity of the ACP-cPNA. Moreover, this sensor was applied to detect the real HBV DNA extracted from human serum samples and compared with the standard real-time PCR method. These results showed that the proposed device can be detected and quantified from the extracted DNA sample without requiring additional DNA amplification methods. And the quantity of HBV DNA was in good agreement with the standard real-time PCR method with a percentage error of 3.50 or less. Here is a quick recap of my presentation. We presented an electrochemical platform for HBV DNA detection with outstanding analytical performance in terms of ease of use, rapidity, sensitivity, and selectivity. The DNA sensing platform can serve as an excellent POCT prototype for other biological assays.